Good evening, everyone. It's a pleasure to have you join us. I hope this session is very informative. We have two very skilled researchers and clinicians, Dr. Jad Chahoud and Dr. Brandon Manley. Dr. Chahoud is going to start off with a presentation about the medical oncology aspect of kidney cancer. And Dr. Manley is going to follow up with some aspects of the research that he's doing. First and foremost, it's important to know that the content is not intended for medical advice, and viewers should contact and consult their physicians should they have any questions. They should not rely on information contained in the presentation for immediate or urgent medical care. And if you think you have a medical emergency, you should call your physician, go to the nearest emergency room, or call 911 immediately. Please don't disregard professional medical advice or delay seeking care because of information contained in this presentation or webinar. So without further saying, let me introduce Dr. Jad Chahoud. He's a GU medical oncologist focusing in kidney and testis and penile cancer. He is doing amazing research in kidney cancer since he joined us after finishing his training at MD Anderson. So go ahead, Dr. Chahoud.
Thank you, Dr. Pow-Sang, for the introduction. So basically, we're going to try and I'm a medical oncologist with some translational interest in kidney cancer research. So we're going to try and go over clinical trials in kidney cancer, starting with 101. What is a clinical trial? It's basically a study that's trying to test the safety and the clinical benefit of a new treatment or a new treatment approach. Why do we do them? Basically to improve early detection of cancers, improve cancer prevention, decrease the risk of recurrence, improve survival, and the quality of life of patients. So basically, I'm going to try and go quickly over how we hope to advance patient-centered clinical trial and drug development in RCC. Next slide, please. So why are clinical trials important for patients with metastatic kidney cancer? Have we improved the survival of our patients with advanced RCC? Yes, we have. Over the past 10 to 20 years, clinical trials have led to the approval of more than 12 to 14 drug or drug combination that have pushed the needle from the 5-year overall survival of stage 4 kidney cancer patients being in the range of 10% now to all the way close to 50% of patients. Did we improve the quality of life of patients with advanced RCC? Yes, but we still have a lot to do, and we can do better. Can we treat advanced RCC that is refractory to therapy? It's a continuous goal at Moffitt with our clinical, translation, and basic science groups. We try to come together to pick and identify potential new target and new mechanism of actions to target RCC. Can we offer clinical trials to every patient in the US? Unfortunately, we're very far from it, but we're hoping with all of our efforts that we can bring more patients, clinical trials here in the state of Florida and in Tampa. Next slide, please. So as you can see, this is the timeline of advancement that we've had all the way from 1992 where high-dose IL-2 was the only FDA-approved drug for stage 4 kidney cancer and where patient 5-year survival was less than 10% and 5%. And unfortunately, that's still something that is very, very common when a patient types on Google, what's the 5-year survival? You may get that old data on your first page of your search. But we've had multiple TKIs that have been approved between 2005 and 2012. And in 2015, we've had the first FDA approval of immune therapies. and in 2018, the first approval of combination immune therapy, which has raised the bar in overall survival. So with two new mechanism of actions, hopefully soon we will have the HIF alpha inhibitor mechanism of action approved in later lines of therapy in the coming month, and hopefully we can move it more upfront. And we're opening trials with this molecule more upfront in the treatment of RCC patients here at Moffitt. Next slide. The second question was, did we improve quality of life? And as you can see on your right hand, these are like some of the quality of life metrics. In red, you can see the quality of life of patient reported on immune therapy combination versus patients who were receiving single agent TKI. As you can see, is significantly lower. But patients who are getting immune therapy combination with TKIs, from a sponsored trial design, they've been receiving the maximal tolerated dose continuously. So we've been trying to work with our great evolutionary and mathematical oncology group to find a way to optimize the drug delivery, the combination of immune therapy and TKI, instead of giving it continuously, giving it in a way where we kind of respect clonal evolution, and we try to optimize the dosing of the drugs to both have maximal longest clinical efficacy while giving patients break from therapy and having time to recover from side effects and have better quality of life. So we've designed this clinical trial, if we get funded by the ACS or we have philanthropic funding, we are ready to get this trial going both at our site and in combination with MD Anderson to accrue patient quickly and move things quickly. If this way of administrating drugs that is already FDA approved can help us improve our therapies and the quality of life of our patient, that's a low-hanging fruit for us as a community to improve patients' life and their quality of life. Next slide. And another more advanced way to finding new mechanism of actions beyond the immune checkpoint inhibitors and beyond the TKIs that are already FDA approved is to try to work on new agent. And as you know, Moffitt is a leader in cellular therapy in general when it comes to TIL therapy, CAR T therapy, or even other novel immune therapies. We have 3 clinical trials that the first 2 we're hoping to open if we get funding. The first one is we have developed through the support of the DoD novel mechanism of optimizing TIL therapy at our Moffitt facilities, specifically for clear cell and even non-clear cell patients, which has never been done before for the non-clear cell patients, never been shown to be able to be done. And we've designed a clinical trial, a pilot study that we will be submitting to DoD for potential funding in patients who have progression of disease after immune therapy. So to kind of have a different mechanism of actions beyond immune therapy to fight kidney cancer. The 2 trials at the bottom are industry sponsored, but they're interesting because the first one is assessing an integrated circuit T cell therapy, basically consistent of autologous T cell genetically modified ex vivo by CRISPR therapy to optimize these T cells using 3 different transgene caskets that has 3 different functional modules. That's one of the probably most exciting cellular therapy trials that we're looking to open in the near future at Moffitt. And we're trying also to repurpose IL-2, which you've seen that was one of the first FDA-approved drugs in 1992 that has limitation because the molecule, the way it is, it causes a lot of side effects. So it's not possible for us to go to high doses to get more efficacy. So this novel way of modifying the IL-2 molecule can allow us to go 100 times higher in the dose of IL-2 without probably less than 10% of the side effects that we see usually with IL-2. So exciting things. The last trial is close to opening and activating at Moffitt. A lot of exciting trials that we look forward to getting supported and open at Moffitt in the near future. I'll move it to Dr. Pow-Sang.
Thank you, Dr. Chahoud. So, as you all can infer, there's a lot of research going on how to improve treatments for kidney cancer. And you probably seen that a lot circles around the immune system. So in this regard, Dr. Manley, one of our surgeons, has been doing a lot of research related to the environment with the immune system. So he's going to give us some insight in the research that he's doing. So go ahead, Dr. Manley.
Thanks, Dr. Pow-Sang. Good evening to everyone. Like Dr. Pow-Sang said, I'm Dr. Manley. I am a surgeon by training, and I spend half my time taking care of patients, and that's what really brings me a lot of joy. The other thing that I really enjoy to do is research, and so the other half of my time is spent doing research, and especially at a place like Moffitt, fortunate to work with people like Dr. Chahoud, Dr. Pow-Sang, along with many other people, including fully dedicated researchers, to try and do better, sort of like the clinical trials Dr. Chahoud mentioned above. There's a lot of things that go into the science and discovery and evaluation of different treatments before they ever make it to a clinical trial. And part of it is just learning more about the cancer. For as much as we know, there's probably a lot more we don't know. So next slide. So fully anticipate nobody here, or maybe some have, know what spatial transcriptomics is. So this is just a slide to introduce the concept. It can be a little bit complicated, but transcriptomics by its nature is just the sort of ability of our cell to generate different genes. We have about 23,000 of them, and many of those genes can get turned on or turned off depending on what that cell is supposed to do. Cells in the stomach or cells in the kidney have different functions, and so they need different genes to do that. But cancer also can behave in a way that's specific to itself, meaning it likes to turn on certain genes or maybe turn off other genes. And that's sort of studying the transcriptomics of those cells, or in this case, specifically kidney cancer cells. Spatial transcriptomics is sort of a newer technology where not only can we tell what genes are being turned on or turned off or overexpressed, but we can tell where they're being done within a tumor. Previous technologies sort of mixed everything together, and we had to use complicated techniques to try and link it back to certain cells, whether that's a tumor cell or like a white blood cell or something like that. But with this technology, we can get both at the same time, and that's really unprecedented in current science. And so this sort of shows at your far left different technologies that sort of fall under this telescope or under this umbrella of spatial transcriptomics. And you can sort of see how, as you go to the right there, you get sort of more and more granular and detailed data. And so this project is using a technology that's very similar to the one on the right called SMI, or singular molecular imaging. It's sort of like having the James Webb telescope at our disposal for the first time, and we're learning a lot more about these tumors. Next slide. You can click the little button. There's a little animation. I apologize. So this is sort of just some of the data that Dr. Chahoud was quoting about some of the trials that have led to some of these novel breakthroughs, which have been really amazing for patients over the last 5 to 7 years. You can sort of just pay your attention to the two big parts where you look at sort of the overall response rate, and you can see across the board, those rates are in the 60s up to even the 80 percentile, which is really amazing for patients, meaning many more patients are responding to it. But what's also important is to notice the CR, which is sort of the complete response rate, where we're sort of shooting for eliminating all the cancer in the body, right? That's where we start to get to use the word cure, which is really exciting for anyone with cancer. Unfortunately, those cure rates or complete response rates are still really low, anywhere from 5 to 15% at best. As we learn more about or have more data on these patients, the longer we use these drugs, that may change, but it doesn't look like it's going to. And so that means while many patients may respond, most eventually will develop resistance. And so this study is trying to figure out how are these tumor cells becoming resistant to these therapies, and how can we maybe take advantage of some of those ways those cells are behaving or changing over time to try and, eliminate them. You can go to the next slide. Next slide. Next slide. There you go. So this is a small study. It was actually started with some donations from one of the patients here previously who really helped us with this, or otherwise would not be possible. So this is actually using just 21 patients, which doesn't sound like a lot and really isn't, but the data gets pretty complex pretty fast with this. And we found through our caring of patients with immunotherapy treatments that some patients had a really good response, and all that we could find left, or at least the bulk of their disease, was left in their kidney. And so at those points, we would go in as surgeons and remove those kidneys. And that's only something we really have been doing over the last couple of years, I'd say. But it also presents a very valuable opportunity for us to study those tumors that inherently, if they are still there after patients are receiving months or even years of therapy, they must have adapted some mechanism to be able to withstand that. So we took some of those tumors to conduct in this study to really study what makes them different, right, compared to tumors before they get any treatment. And so for here, we took actually very small, about 1 millimeter of tumor right in the sort of center part of the tumor, that yellow square or yellow circle, and then right at the edge, that red circle, that's sort of the stroma. And they sort of sit at this interface where we know, especially when it comes to immune system, and specifically the drugs we use in kidney, immune inhibitors, right? That's where the interaction is happening between the patient's immune system and the tumor, right? And there's a battle going on there. And again, looking there is where we have found through previous studies to have the most actionable sort of results. And this is an H&E stain. You can sort of see the purples, the blues, and stuff like that, and get some sense of how things are different. But if you pay attention to this, what I'll show you in a minute, it gets a lot more detailed. So go ahead, next slide. So when we looked at all of these genes that were grouped within the tumors that we took out after patients have gotten immunotherapy versus those who hadn't, we noticed some really big differences. And again, you can sort of turn your attention to the yellow circle there, which is highlighting basically all the tumors. And you can see at the top bar, that purple, that were after immunotherapy versus the ones in the teal, which were before. And the red means sort of up regulation of these genes, and the blue is sort of down. And again, these genes are sort of grouped into different pathways, sort of very basic biological functions, but it's sort of how we can sort of get an estimate of what these cells are trying to do. Are they trying to grow? Are they trying to, become reactive to certain stimuli and stuff like that? But the sort of take-home point for here is that there's many of these genes that are different in the tumors that have been treated versus those that haven't, right? And maybe those can be unlocked to further refine how we, develop new drugs or how we manage with the drugs we already have. Additionally, because of this technology, not only can we look at, all the genes together, whether they're up or down, but we can also see where, right? Is it certain amount of cells within the tumor or in the stroma, like I mentioned? Are they coming closer together, or are they spreading farther apart, right? In order for cells usually to exert their effect, specifically immune cells, they have to be within some distance of proximity. They can't be way out of left field and doing what they're supposed to do, which is, either kill cancer or fight an infection, if they're not within some distance. So that distance, that spatial architecture matters. Next slide. What we have found so far in this study, and it's ongoing, is that not only can we take all of that data we get from this technology, but instead of just doing an H&E, we can actually populate it. And you can see on that right there, a polyploid plot, where we can pick out at a very, sub-millimeter, this is sort of microns, very small distances. We can not only find out which cells are which, right? You can use that key on the right there of all the different colors to know where things like B cells sit, where T cells sit, where tumors sit, where sort of other structures that we commonly find in the microenvironment, like fibroblasts and things like that, where they all sit in relation to each other. And in addition, we can build upon that, and we can see what genes are being expressed within certain types of cells. And there's two genes that have really sort of caught our eye. They're called COLO41A and ITGAV, which are listed at the top. But these ones, when we study them in tumors that have been treated, are dramatically increased. And not only are they increased, but the cells who are expressing them are getting closer and closer together. They mean they have an infinity. And the take-home point is that we think this may hold a fitness advantage for these resistant cells that naive cells do not carry. Next slide. So when we stepped back and found these genes for the first time, because they really haven't been talked about in kidney cancer at the molecular genomic level, we were sort of surprised to see here that when you look, this is sort of listing all the different types of cancer from something we call the Cancer Genome Atlas, or TCGA, that these two genes actually at their baseline are one of the most high expressed in kidney cancer compared to all other cancers. So not only do we think these kidney cancers come in with the ability of having these genes upregulated or making a lot of it, but that may be further enhanced after immunotherapy. And cancer cells, have to adapt to many different stressors in the environment. But one of the big ones, and the one that we care about, is how they adapt to our treatments, right? And this may hold a key as to one of the way these cancer cells are getting around our treatments long term. Next slide. In addition, we followed this up by not only studying the genes, because genes have to get turned into proteins to really exert their effect, but we were able to do this with the protein level, which is called multiplex immunofluorescence, which stains proteins and not genes themselves, right? And we can see that the genes that we cared about, the collagen 4-1A, which is the red one, or the integrin alpha 5, which is the green one here, we were able to also see an enrichment at the protein level, which means not only are these genes these cells making a lot of this gene or enriching it, but they're turning that into protein, which means they're turning it into action, right? So again, this is sort of exciting because now we know that this sort of follows through the whole from starting at the tumor cell all the way into a protein that can have and exhibit an effect, and it becomes a much more suitable target. Next slide. So this is our sort of final hypothesis, at least at this point, where you can see sort of the two receptors I talked about, the purple and the orange sort of dumbbells there, how they're sort of there naturally, right? But after treatment, they sort of come together to exert this effect, which, not to get into the science, but these types of genes can have an effect of inhibiting, or not inhibiting, but enriching fibroblasts to develop around the tumors, which can protect them from a mechanical perspective. But those fibroblasts can also give different stimuli to the tumor cells to help them grow, develop their own blood vessels, and eventually spread throughout the body. This is early stuff. This technology is very early on. There's not a lot of experience. We're very lucky at Moffitt to have experts in bioinformatics, immunology who work with us on this. And there's a lot more to do, right? We're sort of just opening Pandora's box at using this technology. So we have to do it in more patients to really confirm these results. We have to do it in other types of kidney cancer because those are some of the most understudied. And so some of our ongoing projects planned are to do that in things like papillary and chromophobe, where we really have very poor treatment options when it becomes metastatic. So for those patients, it's not studying those tumors after treatment. We need to learn about those tumors before treatment. And then obviously all of this, we can, once we know sort of what switches to pull, so to speak, we can start changing in how we adapt our treatments depending on these, and coming up with either molecular targets or, like Dr. Chahoud mentioned, even immunotherapy targets through TIL therapy, CAR T, and adaptive immunotherapy. Next slide. And that's it. Thank you.
Thank you, Dr. Manley. I want to introduce Mike Quinn, our partner in Philanthropy, who's going to say a few words before going to the question and answer session. So go ahead, Mike.
Thank you, Dr. Pow-Sang, and good evening, everyone. In just a minute, we will return to the Q&A portion of our webinar, which I know is the exciting part of this evening. So feel free to start sending in those questions now, so some of the physicians on the webinar can kind of take a look at those. But I first want to thank you all for your time this evening. I know we all have busy lives and there are so many things that we can be doing on this Thursday night, but you chose to be with us, and we really are grateful for that. I also want to thank Dr. Pow-Sang, Dr. Chahoud, and Dr. Manley for providing such good insight on these exciting breakthroughs. The work they're doing not only benefit local patients, but it has a global impact. I also want to quickly thank some of my foundation colleagues, Brielle Humphreys and Tiffany Hughes, for managing this webinar behind the scenes tonight. And lastly, I hope tonight's discussion was informative and inspiring. There are several GU oncology research projects that are still in need of funding. And if you have any interest in exploring ways you can provide philanthropic support for the important work, please contact me at your convenience. Again, thank you for joining us, and I will now turn it back over to Dr. Pow-Sang to begin the Q&A session. Dr. Pow-Sang?
Thank you, Mike. I want to thank Mike and his team for the tremendous support that makes all this research possible. I also wanted to thank Dr. Chahoud and Dr. Manley for those very nice presentations, very easy to follow. I'm a clinician, but I can understand, so that's a great start. It was short and sweet, if I could use an expression, and it's going to give us a lot of time to address any questions that you have. So I'm looking at the text feed here, and I'm going to start picking up some questions. The first one that I would send to Dr. Manley, Mrs. Filippo is asking if there are any studies going on with papillary kidney cancer.
So thanks, Dr. Pow-Sang, and thank you, Marlene. So the answer is yes, there absolutely is studies going on. It's not so much is there studies, is how many studies, because we need more. Unfortunately for papillary, it's a rarer subtype, so we're only seeing about 10 to 15% of patients. So when you have a smaller group of patients, it makes studying the tumor smaller. This is a problem with many other cancers, not just kidney. We do actually have one that is just started a couple weeks ago through the Department of Defense, where we received money to study specifically papillary patients to learn more about that disease type, because again, the bar is much lower, unfortunately, in trying to learn that. There is other clinical trials, and maybe Dr. Chahoud could speak to that, that are, leveraging for these kind of studies, especially at the clinical trial. You usually have to get multiple institutions involved. So it takes a lot of networking, collaborations, logistics. So there's the clinical trial part. There's certainly a desire to do more, but it takes a lot of effort at the clinical trial level, and at the sort of science or translational level where I sit, it's not always easy. Luckily here at Moffitt, we've done an excellent job of, many patients are willing to give parts of their tumor to research so that we can learn more about them. And so we're thankful for that. And so we've been able to accrue enough that we think we can do some really great studies to learn more about the biology, the nature of them, why they affect some patients, more aggressively versus other ones are very much more indolent. There's many questions to be asked there, and we're starting that process, but I certainly wish and hope we can do more.
Thank you, Dr. Manley. There's a question here.
To add to Brandon's point, and to add to Brandon's input about that, yes, we have. We're also working on papillary and other non-clear cell. We have already generated data that we can grow TIL therapy for those patients. We have designed a clinical trial with using TIL therapy for those patients. Again, the hard work now starts on getting funding to get these trials up and going and making it happen. We have a clinical trial open that's a sponsored study. It's open at probably other sites in the US for papillary patients who have stage 4 disease that has not been treated yet, using immune therapy in combination with new TKI drugs.
There's a question here, and I would ask Dr. Chahoud to approach it. There's a question regarding how much time does it take for a tumor to adapt or become resistant to treatment?
Yeah, so basically, in the frontline setting, we know the data of like time to response, time to develop resistance later on. What we're trying to do with that clinical trial that I alluded to in my conversation with Dr. Garenby and Dr. Anderson from the mathematical oncology and the evolutionary modeling team is to kind of take a patient-per-patient approach, a personalized approach that takes into consideration each patient's response and each patient's MRI imaging to decide when to stop therapy, to prolong that time of resistance. So rather than continuing putting pressure on that tumor, killing cancer clones that are relatively responsive and keeping the fire under the resistant ones and having them grow, by having that adaptive approach of starting and stopping therapy, personalized, dependent on the response of each patient, we hope to prolong that time. So I'm not sure if that answered that question.
Thank you, Dr. Chahoud. There's a question for Dr. Manley regarding if the tissue that has been obtained is used for research, and whether there is a way that patients can help do this research.
So it's a good question. So certainly, like I mentioned, we have an institutional protocol. It's not specific to kidney cancer. It's actually for any patient treated at Moffitt, because obviously we're doing kidney, but there's many great people doing great work in other cancer types too for similar reasons. When patients consent to that protocol, they're allowing us as scientists and researchers to learn more about cancer, whatever cancer that may be. And certainly with kidney cancer, that's what I do, Dr. Chahoud does, along with a whole team of people you don't get to see, but are really working hard. So I can say pretty reliably, if you're somebody who's had papillary renal cell cancer and been treated here at Moffitt over the last couple of years, and you consent to that study or that protocol, the TCC, that we will be utilizing some of that tissue because it's very valuable for some of our upcoming studies, which have now been funded by the Department of Defense. So we're really excited and obviously very grateful for all those patients, because none of this could happen without that.
Thank you, Dr. Manley. It's a question for Dr. Chahoud very early in the question section. Somebody asked, what is the role of statins in the formation of malignancies? Mentioned urothelium, but there's a lot of research regarding statins and prevention of cancer. Can you make some comments?
Yeah, so I don't think it causes, there are some data of using statin in prevention, some data limited using statins in combination with immune therapy and showing that may be a cause of better response, but nothing like slam dunk and that has been recommended to use, or nothing known to cause.
There's a lot of research with statins and prevention of cancer. And there are many agents that are used, not only
metformin, statin has been,
but also different natural agents. There's a question here, a very important question, Dr. Manley. How does a patient know if he's a good fit for a clinical trial?
Well, I'll defer. I think it's important Dr. Chahoud speaks on that. But, there's clinical trials across every part of cancer care, from, the quality of life, which certainly is an important part. In surgery, we tend not to have as many clinical trials as we do once patients develop metastatic disease. Part of that is because surgery, for the vast majority, luckily, we cure a lot of patients. And so there's not to say we can't improve how we care for surgical patients or manage those. But setting up full clinical trials is somewhat difficult. We do have those from time to time. We don't have any active right now on the surgery side. But when it comes to determining on the metastatic side, I think there's a lot of important factors that need to come in. One is your understanding of what a clinical trial is done for. And I think Dr. Chahoud did an excellent job of making that point earlier. And two, it's looking at the current options, or what we call standard of care. For patients where the standard of care does a very good job or has a chance of curing you, which is the case in a lot of other cancers, not a lot, but some, I'd say, then sometimes a clinical trial may not be focused on cure, but focused on the quality metrics or the toxicity of that, how we can make things better while still not sacrificing cure. Many of the trials, I think, in kidney cancer, especially in the metastatic setting, are done to try and improve the cure rate. Now, obviously, they all have a focus on quality of life too. But unfortunately, in kidney cancer, we haven't met that bar of curing enough patients where we can be satisfied. And so for patients, it depends on where they sit in their cancer journey. Have they already had a treatment? What type of disease they have, like we got to the histology, those factors all come into play. For patients without clear cell histology, as part of the NCCN, they actually recommend those patients be considered for a clinical trial, because right now our data on how to best treat those patients is severely lacking. We need to do better. And enrolling patients to clinical trial is still the best way we know to get to that next level. But I think Dr. Chahoud can comment a little more.
Yeah, and I think Dr. Manley answered this perfectly. The thing that I kind of tell my patients and tell every patient with cancer in general, especially stage 4 cancer, is at every decision point of like, what are we going to do next? Clinical trial should be a question. Like, do we have clinical trial options? And then if you have a clinical trial option during that decision, then comes the point of having an informed decision of the pros and cons. So it's part of the informed consent that you will be signing for any clinical trial, is that your doctor needs to explain to you the standard of care therapy options that are available first, and they need to offer you that first. And then you have to make a conversation and this discussion with your physician at that time point, like if that trial one will add any benefit, if he believes it will add benefit. 2, if you're an eligible candidate, which you will go through screening testing, and 3, if how can it work for your situation, having to come to Moffitt, having the logistic of a trial that will add. And at every decision time point, it should be a discussion if it's an option for a patient, because it's like a tool belt where we're trying to add tools every time, and a clinical trial is like a potential additional tool.
Thank you, Dr. Chahoud. Question for Dr. Manley. What are the survival rates for stage 1B renal cell carcinoma after nephrectomy? Where does it metastasize? The most likely area to recur, and what treatments would be available in that situation?
Yeah, so it's a good question. So stage 1B is actually a fairly good prognosis. The lowest stage you can have after surgery is stage 1A. So stage 1B is just up from that. If you look, there's several different calculators that are out there. There's the SURE trial, which has probably the latest and greatest when it comes to this. There's some other ones to calculate sort of your estimated risk, but roughly, if you're assuming the patient had clear cell, which the histology does matter too, the cure rate is close to 90% for those patients over 5 to 10 years. Now, for patients who had a chromophobe or something, which tends to be more indolent, it's probably higher than that. Versus somebody who had a more aggressive type of like the papillary type 2, it is probably lower than that. And then, there is rare kidney cancers that we don't see too often that really don't play by the rules, things like medullary collecting duct, where those are, unfortunately, they're only about 1% of patients, but for those patients, they can be very aggressive, independent of the stage. So those are the big things. When kidney cancer comes back, the most common spot it goes to is the lungs. So you may find, especially seeing me as a surgeon, we always surveil or get an imaging of the lung before so that we have that as a baseline. And for patients who have metastatic disease, see Dr. Chahoud, that's part of what they monitor as well. The lungs is the most common. After that, it's usually the adrenal gland, the lymph nodes, and then the bones tend to be sort of the ones that follow suit after that. But kidney cancer can go a lot of unusual places as well. So that's not by all means definitive. When it comes to treatment options, then it really gets into what Dr. Chahoud and his team does, and it's usually systemic, but there is exceptions to that. Metastastectomy, which is where we remove a tumor that is spread outside the kidney, does have a role in kidney cancer. It depends sort of, again, how fast it came back within a couple months versus sometimes a couple years, because that gives us insight into the biology of the disease. And it's been well established for patients who only have one site or a site that could be resected, meaning usually surgery, that those patients can have extended survival rates for undergoing that. But, it gets more probably to the biology of that person's tumor than so much of our treatments. But there is debate in that. So, systemic therapy is usually the first option, but there is certainly a time and a place for additional surgery for those patients, even if it spreads. I think you're on mute, Julio.
Dr. Chahoud, thank you, Dr. Manley. A question for you, Dr. Chahoud. What are your thoughts about early detection techniques with circulating free DNA, what are called blood biopsies? Do you think it has an advantage, a benefit with early detection versus doing overdiagnosis?
Yeah, so that's a very smart question, and the kidney cancer research community has been trying to tackle that one for the past decade. We're not there yet. The ctDNA technology for kidney cancer is not at the level where it can be FDA approved and used for early detection, but I think that's going to be probably the future. Maybe something that will be coming sooner is zirconium PET CT, and Dr. Manley can probably talk more about that because earlier studies have been used for small renal masses, and I think that probably will be in the future of the management of small renal masses.
Yeah, I'll make one comment. I think you're right that that study is recently concluded, and I think they're doing follow-up. So many of us in the community are eager to have, like Dr. Chahoud mentioned, another tool on our tool belt. To speak about liquid biopsies, like Dr. Chahoud mentioned, that's been sort of, I don't want to say the holy grail, but we all want to do that because there is some cancers like prostate, other ones where we have the ability to get a much better picture of what's going on from a cancer perspective with a simple blood test. We actually have a program here through a grant through the state of Florida through Bankhead, where for many of the patients who have consented to TCC, we've been collecting blood samples before and after surgery, and for some of Dr. Chahoud's patients, before and after they start systemic therapy. But unlike DNA, we've actually been looking at specific splicing in RNA. And again, it's a novel, totally different way to do it. It overcomes some of the challenges of looking at DNA, which can be very non-specific, but it has its own challenges. RNA is not nearly as stable as DNA, and so the techniques that are needed in the lab and an analysis can be more complex. But it's certainly something we're trying to do here and have the support of the state and obviously patients to do that. And that's an ongoing study. And again, another way that the patients who donate or allow us to do that are really helping us try and look for those keys, because it would be a dramatic breakthrough if I could order more blood tests for my kidney cancer patients versus all the scans we unfortunately have to do at this point. I think that would be a win.
Thank you, Dr. Manley. Dr. Chahoud, a question about stem cells. Do they have any application at present? Any insight about that?
Currently, no. Basically, TIL therapy is the closest using your own immune system. And basically, it's a way of trying to collect T cells from patients' own tumor and identify the T cells that are doing a good job and grow them outside of your body in plates to multiply to the billions and then reinfuse them to the patient in hope of having these T cells now that are much higher quantity, being able to overcome the challenge of beating the cancer cells. So that's probably the closest that we are to using the own patient's immune system. We have an autologous CAR T trial, and then that other trial that I've highlighted that also uses autologous, not your own T cells that have been genetically. So the newest trial will genetically engineer these T cells using like CRISPR technology, where they can put modify these T cells to be kind of stronger and smarter, and then give them to patient in hope of overcoming cancer.
Thank you, Dr. Chahoud. as these presentations and this session is focused on kidney cancer, but there are two questions on bladder cancer. So I'm going to ask Dr. Manley if he could address them. One is related to the use of blue light cystoscopy. Can you comment on that as it relates to bladder cancer?
Yeah, so blue light cystoscopy is very specific to bladder cancer, has no role or place in kidney cancer. So to start with that, blue light cystoscopy is sort of a technology that's actually been around for about 10 to 15 years now. And the idea behind it is that when we do a typical cystoscopy, for those who don't have bladder cancer, that involves us putting a small camera in through the urethra into the bladder, because that is our most sensitive way to pick up bladder cancer, especially when we're monitoring for recurrence. But that depends on classical white light, the light that we're all used to. Blue light, which again is, I'd sort of give the equivalency if people, you watch like the Predator or Terminator and they switch into, like infrared, right? This is sort of doing different shades of light, because what this technology does is they infuse a chemical basically into your bladder before, and that chemical gets soaked up more by cancer cells than normal cells. And if you put the right light beam on it, those cells, actually, that chemical will start to flash red, basically, or turn red. And so it makes it much easier, in theory, to pick up smaller tumors or difficult to locate tumors than traditional white light.
Thank you, Dr. Manley. There was a question here related to how one could find more information about research being done at Moffitt. And one very good resource that keeps up to date is going to the website, moffitt.org. If you look at the screen at the top, you're going to see clinical trials, and you can go by organ site and then get more information. Also, a contact number that you might call directly if you have more specific questions through that website.
I would also mention, I think Michael can provide some of that too. We have briefs on all of in GU, and I'm sure all the departments. So Michael can be an excellent resource for those kind of up-to-date projects as well.
That's right. Good point. There's a question here regarding mathematical oncology, a concept called adaptive therapy, in which you don't use maximal treatment, but you kind of cycle the treatment or modify it to delay resistance. Can you make some comments about that, Dr. Chahoud?
Yes, of course. And it's a very smart question, whoever asked it. And basically, in our trial design, that trial that we're hoping to get going, I made sure to include our patient advocates to have their input on the trial. And one thing that we did is the trial allows the patient, and we're going to assess the acceptability. So patients are going to be offered the trial if so at the beginning, once they start therapy, before knowing if they're going to respond or not. And then they're going to, at the time of where we think they should stop based on this adaptive approach, they're going to be offered the opportunity to either continue continuously with like the old methods, or stop and then follow and then restart. And that's the goal of that, is to assess the acceptability, because this approach has not been tried, as you would think. When these studies are designed by sponsors, the thought of stopping therapy is not going to come to anyone's mind who's designing the trial. So it has not been looked at before. We believe that it will have a good acceptability. I believe it will allow us to have these drugs that work longer in patient's body and hopefully work for longer.
Thank you, Dr. Chahoud. Question for Dr. Manley. Is there any hereditary connection to kidney cancer? And are you seeing an increase in incidence in younger people?
So there is definitely a hereditary component for some kidney cancers. We estimate it to be about 5%. But if anything, that number will only go up as we learn more about what causes kidney cancer, especially when you inherit genes from your mom and dad that can predispose you. The most classic syndrome we talk about is von Hippel-Lindau. It's sort of been around and well established, but there is other ones, hereditary papillary, Lynch syndrome, and some other ones that we've more recently learned more about. This is an expanding field. For patients who have hereditary predisposition and have either developed kidney cancer or are at risk of developing, we treat those patients very different than we treat the normal population. So for younger patients, and we sort of use the cutoff of 47, but there's other risk factors we include. But if you're under 47 or you have a strong family history of kidney cancer, that sort of raises our suspicion for that. And so for those patients, we get them in touch with our genetics team, who do a really excellent job of thoroughly going over your family history, talking about some of the benefits of genetics and some of the risk, right? But having that information either before your treatment or after your treatment will allow us to modify and treat those patients differently, not just with potentially systemic agents for those who are metastatic, unfortunately, but also for those who have localized disease. We may monitor those patients the rest of their life. And they may also be at risk of other tumors, right, that they didn't know. Kidney cancer may just be the first one that showed up. So those are important patient populations. They're not as the bulk of patients, but those are definitely the patients that benefit the most from that. Unlike some of the other cancers like colon cancer and stuff, where we've seen a shift towards a younger age population, we really haven't seen that with kidney cancer. There's been an increase in kidney cancer overall when you look at the last several decades, but most people believe that's actually because us in medicine are using way more imaging scans than we used to use 20 years before, right? You don't walk into an ER basically now without getting a CT scan. So the incidental detection of kidney tumors or masses has gone up, but it's pretty much gone up across the board for patients, not just one population.
Thank you, Dr. Manley.
And to add
Go
to
ahead,
Dr.
Chahoud.
To add to Dr. Manley's great comment, and Dr. Manley sits on the NCCN guideline for genetic hereditary syndrome of RCC, and we at Moffitt also are part of the VHL Alliance leading centers, where it's one of the most common genetically inherited syndrome in RCC, and we have a multidisciplinary team between neurology, neurosurgeons, and everyone that need to take care of these patients is available at Moffitt to give them the best care possible.
Thank you, Dr. Chahoud. There's a question here regarding if somebody has lost one kidney and then the bladder is involved, is it more likely that the cancer will spread to the other kidney?
So I can answer that. I'm sort of making some assumptions here, but what that situation is more likely describing is urothelial cancer than traditional kidney cancer. When we say kidney cancer, and this can get confusing for a lot of patients, we're usually talking about tumors that originate from sort of the meat on the kidney, or what we call the parenchyma, the cortex. The inside lining of our kidneys, though, what we call the renal pelvis, is actually the same lining of cells that lines our bladder. So in theory, patients can get what is more equivalent to bladder cancer up in their kidneys, but it is not kidney cancer. But in the situation where you assume they're referencing a urothelial cancer, which can present initially in the kidneys, in that renal pelvis part, the most common spot patients get recurrence after that is the bladder. And so that's part of their monitoring plan. And about, again, depending on the stage and grade of their initial tumor, that risk is anywhere from 25 up to 50%. The incidence of tumors developing on the contralateral, or the other kidney that's left, again, depends a lot upon the stage and grade of their initial one, but it's quoted from anywhere from 2 to 10%. So it's not as common, but it does happen in those situations. When you only have one kidney, become a very different and sometimes difficult situation to manage. So again, it just highlights the need for continued and close surveillance, which for many of those patients is pretty intense the first 2 years. But the idea is to catch things before they get big, and that maximizes the treatment options we have available.
Thank you, Dr. Manley. Well, it's amazing. We addressed all the questions and we're 5 minutes ahead. So I want to thank Dr. Manley, Dr. Chahoud for their great presentations, and Mike and his team for putting this together, and for you all to participating in this webinar. It's always a pleasure to share all the exciting research that we're doing here at Moffitt. We're always available. You have our contact information, so please reach out to us anytime. Thank you and good night.